Ever wondered how we measure pH or electrode potential? Today, it's all about potentiometry. Let's dive right into it. Potentiometry, in the simplest terms, is a method used in electrochemistry. It's like the detective of the chemistry world, helping us measure the electric potential of a solution. This might sound a bit technical, but think of it like this. It's a way for us to uncover the hidden secrets of a solution, to really understand what's going on at a chemical level. Why is this important, you may ask? Well, potentiometry is a key player in a variety of fields, not just chemistry. For instance, in biology, it's essential for understanding how cells work and communicate. In environmental science, it helps us track pollution levels and the health of our ecosystems. So you see, potentiometry doesn't just live in the lab, it's out there in the world making a real difference. But how does it work? Well, potentiometry involves the use of electrodes, a kind of chemical sensor, which are immersed in the solution we want to study. These electrodes measure the potential difference, or voltage, between themselves and the solution. This difference in voltage can tell us a lot about the solution, such as its pH level or the concentration of certain ions. There are different types of electrodes used in potentiometry, each with their own unique roles. These include reference electrodes, like the normal hydrogen electrode or the silver-silver chloride electrode, and indicator electrodes, like the glass or ion-sensitive ones. But don't worry, we'll delve deeper into these in the next scene. So in essence, potentiometry helps us peek into the chemical behavior of different substances. It's a powerful tool that, despite its complex name, is really all about helping us understand the world around us, and inside us, a little bit better. To understand potentiometry, we need to delve into its theory and principle. At its core, potentiometry is all about measuring the potential difference between two electrodes, a reference and an indicator electrode. This potential difference arises due to the selective ionic activity of the indicator electrode when immersed in an ionic solution. Now, think of it in terms of a balance scale. Just as the weight on one side dictates the tilt, the ionic activity of a solution influences the potential difference. But here's the twist. This relationship isn't linear. Instead, it's proportional to the logarithm of the ionic activity of the solution. This logarithmic relationship is derived from the Nernst equation, a fundamental equation in electrochemistry. It means that even minute changes in ionic activity can lead to significant shifts in potential difference. This principle is what makes potentiometry a powerful tool in electrochemical measurements. Now, let's talk about the actors that make potentiometry possible, the electrodes. These are not your ordinary pieces of metal. They are specially designed to help us measure the potential difference in a solution. And there are two main types of electrodes at play: reference and indicator electrodes. Reference electrodes, as the name suggests, provide a stable point of reference. They maintain a constant potential regardless of the solution they're dipped in, and this stability is what makes them so important. There are several types of reference electrodes, each with their own unique characteristics. The normal hydrogen electrode, for instance, is the standard by which all other electrodes are measured. Even though it's not commonly used in practical applications due to its complexity, it's still our primary reference point. Then we have the calomel electrode, a widely used alternative to the normal hydrogen electrode. It's known for its stability and ease of preparation, which makes it a popular choice in many laboratories. The quinhydrone electrode, on the other hand, is used specifically for measuring pH in slightly acidic to neutral solutions. And lastly, we have the silver-silver chloride electrode, another widely used reference electrode because of its predictable behavior and ease of use. Now let's move on to the indicator electrodes. These are the stars of the show, the ones that do the actual measuring. They respond to specific ions in the solution, and their potential changes depending on the concentration of these ions. A notable example is the glass electrode, which is most commonly used to measure pH. It's a type of ion-sensitive electrode, and it's incredibly sensitive to hydrogen ions. And that's our lineup of electrodes. Each one, from the normal hydrogen to the glass electrode, has a crucial role to play. They all come together to make potentiometry the powerful tool that it is. These electrodes, each with their unique properties, are essential in the world of potentiometry. How do we actually measure electrode potential and pH using potentiometry? Let's find out. First, let's explore how to measure electrode potential. It starts with a potentiometer, which is essentially a tool that measures the electrode potential difference between two electrodes. To do this, we connect a voltmeter across the two electrodes in question. The voltmeter then measures the potential difference or voltage between these two electrodes. This is the electrode potential. But what about pH? That's where a pH meter comes in. A pH meter is a device designed to measure the acidity or alkalinity of a solution, which we refer to as its pH. The heart of a pH meter is a glass electrode that is sensitive to hydrogen ions. When the electrode is immersed in a solution, the hydrogen ions in the solution interact with the glass, creating a potential difference. This potential difference is proportional to the concentration of hydrogen ions in the solution, which gives us the pH. Now you may be wondering, how can we be sure that these measurements are accurate? That's where calibration comes in. Calibration is the process of adjusting a device to ensure that its readings are accurate. For a pH meter, calibration involves testing the meter with solutions of known pH values, called buffer solutions. The meter is first immersed in a buffer solution with a known pH. If the meter's reading matches the known pH of the solution, then it's accurate. If not, the meter is adjusted until it gives the correct reading. This process is repeated with buffer solutions of different pH values to ensure the meter's accuracy across the entire pH scale. With these techniques, we can accurately gauge the electrode potential and pH of a solution. Potentiometry isn't just about measuring potential and pH. It has far-reaching applications, including something called potentiometric titrations. Let's dive in to understand this concept. Potentiometric titration is a technique used to determine the concentration of an unknown solution. Here, the potential difference between two electrodes is measured as one solution is titrated into another. This method is different from other titration methods because it doesn't require a color change to detect the endpoint. Instead, it uses the change in potential difference to pinpoint the precise moment when the reaction is complete. Now, you might be wondering, where do we apply this technique? Well, it has a wide range of applications. In the pharmaceutical industry, for instance, potentiometric titration is used to determine the concentration of active ingredients in drugs. It offers a high degree of precision and accuracy, which is critical in drug formulation. In the food industry, this technique is employed to determine the acidity of various products, such as juices and vinegars. It helps ensure consistency in taste and quality, and aids in meeting regulatory standards. Furthermore, potentiometric titration is a valuable tool in environmental monitoring. It can analyze water samples to measure the levels of specific contaminants. This information is essential in assessing water quality and identifying potential health risks. But that's not all. This technique is also used in the field of materials science to study the behavior of certain materials under different conditions. This information can help in designing materials with specific properties for various applications. In essence, potentiometric titration, as a subset of potentiometry, plays an integral role in a multitude of fields. It allows us to quantify chemical reactions accurately, helping us to understand and control them better. Potentiometry, thus, is a crucial tool that aids us in understanding and interacting with the chemical world around us. So, that's a wrap on our journey through potentiometry. We've navigated the highways and byways of this fascinating field, from its basic concept to the intricate principles that govern it. We've explored the intriguing world of electrodes, each with its own unique characteristics and uses, from the humble hydrogen to the complex calomel, the quirky quinhydrone, and the sterling silver chloride. We've delved deep into the different types of indicator electrodes like the glass, ion-sensitive solid, liquid, and gas membranes. We've also shed light on how electrode potential and pH are measured, and the crucial role of the trusty pH meter. And finally, we've looked at potentiometric titrations, a technique that has its roots in potentiometry and is used in a myriad of applications. So keep these key points in mind as you continue your journey in chemistry. Subscribe for more because you won't find me later. Until next time, keep exploring the fascinating world of chemistry.